In this video, we're going to discuss cell death from chapter number 4 of Pathology at Glance. Let's begin. Starting with apoptosis. Apoptosis, often called programmed cell death, occurs during embryological development as new tissues are formed and remodeled, or physiological cycles such as the menstrual cycle. So, it is a programmed cell death. Apoptosis is characterized by orderly breakdown of cellular constituent which are packaged into membrane-bound vesicles and tagged for collection by the phagocytes. Two stages of apoptosis can occur in 4 steps and 2 stages. Initiation of apoptosis may be via extrinsic or intrinsic factor. Caspases are the central to apoptosis. So far, 14 have been described. Effectors of caspases are enzymes which cleaves protein in membranes, the nucleus and cytoskeleton, and activate the nuclear DNAs. Let's look at the initiation of apoptosis, which include the extrinsic factor and intrinsic activation. Extrinsic factors includes death receptor, pathway stimulated by cross-linking receptor, for example, TNF secretion by immune cells. Direct activation, for example, granzymes B injected into cells by T helper cells through holes in the membrane made by perforin. Then we have third step, intrinsic activation. This is the main pathway by mitochondrial membrane which releases the cytochrome C and other apoptotic molecules influenced by BCL2 family membrane, for example, growth factor blockage. Let's look at the uh, steps in initiation of apoptosis in detail. Remember, this process always requires energy. Initiation of apoptosis include binding of a death ligand, for example, T cells, NK cells, or necrosis factor. Membrane distribution by perforin, then intracellular injection of granzyme B by cytotoxic T cell, release of pro-apoptotic protein, for example cytochrome C, from leaky mitochondrial membrane, a process largely regulated by pro and anti-apoptotic proteins of the BCL2 family, as we said before. Lastly, TP53, a gatekeeper gene in cell cycle, P53 protein instigate apoptosis if there is a failure to repair DNA damage. Once started, this is generally irreversible, involving a final common pathway of an intracellular cascade of capaces. Proteolytic cleavage of cell content and water loss cause cell shrinkage. Fragments bud off, involved by cell membrane, which expresses new ligands. Apoptosis does not stimulate an acute inflammatory response. Instead, macrophages and adjacent cell bind the new ligands and phagocytize the fragments. Next is autophagy. The body attempts to preserve cells through times of adversity by undergoing autophagy, a particular type of cellular adaptation commonly seen in starvation and in infection. It is also initiated by growth factor deprivation. Portion of cytoplasm are bound by membranes to form a vesicle, autophagosomes, which fuses with lysosome and its content once degraded by hydrolase enzyme and are then recycled. The process is like hibernation and can be reversed once the lean time pass, but if taken to extreme, because the stimulus persists, the cell dies either by apoptosis, as discussed earlier, or necrosis. Pathological examples of diseases in which autophagy plays an important role including neurodegenerative diseases such as Alzheimer's, Parkinson and Huntington's, disease, much interest has recently developed in role played by autophagy in cancer. So this diagram here represents the autophagy process in detail. Next is necrosis. Necrosis is the death of a group of cells due to noxious stimulus, which is referred to as necrosis. It is caused by many physical or chemical agents, amongst the most common of which are ischemia, hypoxia or reperfusion, for example, in myocardial infarction, infection, it can be bacterial infection, viral or fungal, immune mediated category involved cross-reacting antibodies, physical agent, for example, extreme heat or cold, electrocution or irradiation. Chemical agents can include strong acid or alkali drug which binds DNA. Nutritional factors include folic acid, B12, vitamin deficiency. It has been suggested that necrosis is what happened after the cell death, which mean it is irreversible damage, and that changes up to this point, which can be reversed, should be classed as the cell damage. The point of no return is best recognized when there is a loss of membrane integrity and influx of calcium into cytosol from interstitial fluid or from the endoplasmic reticulum. So let's look at what happens in the process of necrosis. Starting with sodium potassium pump in cell membrane, it will fail. For example, loss of ATP production by mitochondria. Lactic acid may accumulate. Sodium ion enters the cell passively accompanied by water. Then the cell will swell. Calcium or magnesium pump will fail. Excess cytosol calcium ion will activate enzyme which depletes the ATP. The membrane bulb and ribosomes dissociate from endoplasmic reticulum. Cytoskeletal components clump. This situation is still reversible. Enzyme activated by calcium ion digests the cell membrane cytoskeleton and the nuclear chromatin. This damage is then irreversible. Various nuclear changes may occur. It may shrink and get darker, which is called pycnosis, fade away, karyolysis, or fragment chirorexis. Similar nuclear changes are seen in apoptosis, particularly chirohexis, which is formation of fragment. Some claims that this appearance is unique to apoptosis, but it is disputed. Release of cell content stimulates acute inflammation. This adds to the damage by releasing further digestive enzyme and free radical into the tissue. If proteolytic digestion predominates, there is less inflammation and the result is coagulative necrosis. If hydrolytic enzyme digestion predominates, the tissue will undergo liquefaction. Factors that influence whether the damage is reversible in necrosis or not. Let's see the factors. Number one is duration of stimulus. For example, ischemia due to coronary artery thrombosis will cause cardiac infarction, but if the occlusion is rapidly cleared, the area of cardiac muscles that dies will be reduced. Reperfusion may cause problems due to the release of free radical in reprecursed territory. Number two is the dose of chemical agent. What can cause cell death in some people does not in others due to genetic polymorphism, variation in inherited gene encoding, the liver enzyme that metabolizes the drug. The tissue type and its metabolic activity, the neuron of the brain and cardiac muscle cells are highly metabolically active. Energy production from glycolysis via anaerobic pathway is available for the liver liver muscles which store starch but produce toxic lactic acid within the cell. Damage begins within minutes in brain, but striated muscles can be deprived of oxygen for several hours. Cooling of the tissue reduces their metabolism and increases the survival time. Number four is the state of health of existing tissue. For example, iron overload in hemochromatosis renders the liver more susceptible of damage by other toxins like alcohol. So these are the factors that influence whether the damage is reversible in necrosis or not. The number one was duration of stimulus, number two dose of chemical agent, number three tissue type and its metabolic activity, and number four is the state of health of existing tissue. Next is free radicals. Free radicals are highly reactive anion with an unpaired outer orbital electron. They react with inorganic or organic chemicals to form further free radicals. Important examples are reactive oxygen species, hydrogen peroxide, superoxide anion radical, hydroxyl radical, nitric oxide, and so on. Formation of free radicals. During the normal cellular energy generation by oxygen, reduction and electron transfer, killing of pathogen by phagocytes ROS are performed in the membrane complex, unwanted byproduct of intracellular oxidase reaction. Radiation, example generate hydroxyl and hydrogen free radical by ionizing water. Toxic byproduct of drug chemical metabolism by cellular enzyme, for example in the liver. These are all the 4 or 5 types where radicals will formed in your body. Harmful effect of free radicals. Lipid membrane damage by pre-oxidation affects the cell membrane and the membranes of organelles. Protein damage by amino acid oxidation, cross-linkage of protein breakdown, for example microtubule aggregation, and DNA damage can cause mutation and cancer. Protection by free radicals. Natural decay to oxygen and H2O2, which is your hydrogen peroxide. Antioxidant, for example, glutathione and vitamin A, C, and E. Binding of copper and iron to transport proteins. Scavenging enzymes that break down H2O2, again, hydrogen peroxide, and the superoxide anion, for example, catalase, superoxide, dismutases, or glutathione peroxidase. These are all the free radicals which will protect your body. Bring us to the end of today's lecture. Thank you for watching. If you like my video, don't forget to subscribe for more.